Okay, welcome, Bio 11. Oh, hi, little buddy. That's little buddy right down there. It's coming in to say hi. So, hi there, Bio, not 1140, sorry, 304. Getting ready for our last test of the semester. And yeah, you guys have been a really good class. I've really enjoyed this whole semester. And I'll see some of you in astrobiology this fall. That'll be a really fun class. Really looking forward to it. I mean, where else do you get to teach about aliens? So I know a lot of you were asking about a review, and yeah, this is it. It had to be tonight. Sorry about that. Thanks for understanding. Last night fell through. Things happen. But at any rate, it's May 3rd. Tomorrow is May 4th. I've already got my Star Wars shirt going here. But what I want you guys to do is you are welcome to post any questions you have in the top chat. And I've got the test in front of me to make sure that we have exactly what you need, so I don't lead you astray on questions and stuff you don't need to focus on. So if you've got specific questions from the study guide, ask me, and then the best way to do that is don't cut and paste the question from the study guide. That makes me cranky, to be honest. Ask me something about ask me the question, tell me what you're thinking, give me something about where you're going with the answer. And, sometimes you're completely lost, and that's okay. It's fine. Just give me something to bite on to talk about it. But until I get some questions, let me just give you guys some help really quick. You guys deserve it. This class is hard. There's a lot of material, especially here at the end. It's super beautiful weather here in New Mexico right now, and it's hard to study. I get it, and this is the night before, so let me help you out here. Plants and animals are multicellular organisms. List the features they share in common as multicellular organisms. Just remember, multicellular, right? You got to have differentiated tissues. You've got cell differentiation going on. So they're all the cells of a multicellular organism, whether you're a fungus, a plant, or an animal, they're all clones of each other, right? But those cells differentiate into different tissues. Tissues form organs. Organs form organ systems, right? And then not only do you have organ systems, that forms the organism. And all those cells have to be coordinated. They have to talk to each other. So there has to be a transfer of information. And there has to be a transfer of nutrients and minerals throughout it. Okay, so that's question number one right there. So I'm getting some questions rolling in here. I just want a clarification on 45 and 51. I have no idea what 45 and 51 are. I don't have the study guide in front of me. I got the test in front of me. Let's see what we got here. I could probably pull it up. Let's see here. Study guide. Yeah, when you ask me questions about that, I don't even know where the study guide is here. I put it in here somewhere. Yeah, just I don't know what questions 45 through 51. I write those questions as I go through the PowerPoints and through the book chapter. So you want to ask me something specific about them, go for it. All right, now, okay, regarding countercurrent, what would happen if the blood flow was in the same direction as water flow? That is a fantastic question. Think about it this way. If you're flowing in the same direction, you're going to have oxygen-rich water hit oxygen-rich or oxygen-depleted blood. It's going to be a huge gradient. Oxygen is going to quickly diffuse into the blood. However, it's going to reach equilibrium about halfway through, which means there won't be any more diffusion of oxygen into the blood. So you will get less diffusion than if it's going in opposite directions, where your most oxygenated blood is reaching your most oxygenated water first. That's good because there's still more oxygen in the water than in the blood, even the blood's oxygenated. So you get diffusion across the entire gill. It never reaches equilibrium. Okay. Information about the last day of slides. I'm sorry. The one thing I told you guys about that was, be able to list the census and whatnot. Oh, where did it go here? I've already lost too many things open at once here. Yeah, ask me a specific question. I mean, like I said, I was going to go through this here. Animal census. Yeah, animal census are on there. I told you guys. You need the list of environmental stimuli detected by animals. I think that's about it. I'll get to that in a bit. I'll get to that question here. Alexander, you're fine. You're trying to figure out what, because I told you in class, like I wasn't going to have much on there. So that's a totally legit question. It's not a bad question. You're trying to get at you're trying to clarify what I was saying in class. So don't worry about it. It's just, I don't have that study guide in front of me. And I'm sitting there looking for it. And I'm like, where did oh, there it is. Study questions part 2. I was looking at it. All right, here we go. I can help you out here. Yeah. Oh, I don't even have them on there. Part 2. I don't have 45, 41. Huh, I may have added it and forgot to change it. Yeah, make sure you know those things about Ampulla, Lorenzini on sharks, and really know something about the 5 types or 6 senses that you guys that I listed in class. That's it. You don't have to worry about any of the other stuff, especially action potentials or anything like that with the senses. Definitely gonna have to know about action potentials though. All right. Okay, let me go back up to here. I've got the test in front of me. I'm just kind of going through this to make sure you guys know what you need to study here. Another question. All right, 250 million years ago, there was a really bad extinction. It wiped out all the synapsids for the most part. Not all of them. I mean, it wiped out like 90% of marine life, 75% of all terrestrial life. I mean, it was bad day on Earth, right? In the Permian, if you went back to the Permian 255 million years ago, the synapsids, that's the ancestors to mammals. They were dominant. They were the dominant land vertebrates. Jump ahead. After that big extinction, the synapsids are tiny little things. But it's the archosaurs that begin to dominate. And the question is, what changed in the Earth's atmosphere during the Permian? There's a chart on my PowerPoint. Make sure you go look at that. There's something big that changed. There's a lot less of the stuff that we really like to breathe in. But the archosaurs clearly had a different lung than the synapsids that almost certainly gave them an advantage. Something about one lung in a synapsid going 2 way and the archosaur going 1 way. Why would that be important 250 million years ago? That's question number 2. Counter-current exchange. You already asked that question. I just explained it. There you go. Yeah, how does it maximize oxygen absorption? Here's a good question for you guys. This is a definition style question. And you guys have all had cell and molecular biology. You know that there's cellular respiration, which, as you know, your teacher loves cellular respiration. I think it's just utterly fascinating, especially the whole chemiosmosis making most of the ATP production. But that is cellular respiration at the cellular level. And then when we get to studying animals, even plants, there is respiration from a physiology point of view. What's the difference? One is about, of course, making ATP. One, of course, is about moving gases, oxygen, carbon dioxide. And make sure you look those differences up. ChatGPT is did I get that right? GPT. That's a really good place to kind of go for that one. And if you use ChatGPT on your answers, if you're just got chat up and you're taking all these questions in there and posting them in there one at a time to get your answers, I'm not saying don't do that. And in fact, it's a great way to like figure out some stuff that's hard. But we will know if you're just giving a generic answer from ChatGPT or if you're giving stuff that's in our textbook and specifically things that I talk about in class that is not in the textbook or the chat won't give. So you might memorize every one of these answers from chat, right? And you still might not make an A because I have stuff in there that's not in chat. And I know this because I use chat too. So I see what kind of answers it's giving me. All right. Naya, how in depth would you like us to go the differences between sympathetic and parasympathetic nervous system? Do you want an A or a B? I mean, you need to really know like what they're doing. So yeah, I mean, every time I get a question about how in depth, the question really comes back to you. On a question like that, that's pretty big. Yeah, I would know it. Definitely, know that like the parasympathetic, is and the sympathetic. Know those differences, right? They're both of the efferent motor division. They are autonomous, so they're you don't think about it, right? You don't think about I need to release adrenaline right now and get to the choppa, right? Because I'm being hunted by an alien, a predator. You you need to know like what happens during the parasympathetic nervous response and what happens during the sympathetic nervous response. One is amping you up. There's a whole lot of physiological changes that take place, from dilating your blood vessels to increase heart rate, to increase release of glycogen into your blood, to stopping of sorry, it's late at night, stopping of digestion. And then when you go back, and then you had the rest and the relaxation, right? And your body kind of returns back to normal. Digestion picks up, and your blood vessels may constrict a little bit. Okay, Adam, green snarky 8. I like that. Why does a neuron fire in one direction and is all or nothing? That's pretty cool. That's a good question. When a neuron fires, and what happens is it's sending an action potential down the axon. That's a neuron firing, right? And whenever it starts, it reaches a threshold, and that threshold, depending on the axon, depending on the neuron, the animal, where it's at, is right around minus 55 millivolts. And what's happening is you've got these voltage-gated sodium channels. And the reason why there's a threshold is because for that gate to open, the activation gate to open like this, you must reach minus 55 millivolts or whatever that threshold is. Once you do that, that gate opens, and they all open at that particular node, and they go whoosh, just like that, and all the sodium ions flush in. And what that does is that depolarizes the membrane. You go from minus whatever your resting potential is, minus 70, minus 90, minus 65. It varies. But you go from that resting membrane potential, minus 70, and you go to plus 5 as the positive sodium ions rush into the axon of a cell of a neuron. It's all or nothing, because once you reach that threshold, all the sodium gates open up, all the sodium floods in, and your action potential goes all the way to plus 55, and then that will travel right down the axon. And every time it travels and opens up those gates, I'll get you ice when I'm done, okay? Can you wait? I'll get you some in a minute, okay? Wants ice cream. That's what she's asking for, is ice. Can you wait? You have to wait. I'm in the middle of something. Hold on. Class, this will not end, and I'm going to have to give in to the demand, or else this will not end. Maybe that's why it never ends, because I give in to the demands. Parenting 101. There is no right way to do it, and I'm just another parent. That's an AT-AT exoskeleton. Let me finish it. Okay. So the reason why, Adam, that it's all or nothing is because once that threshold is met and the sodium channels open, that floods the axon with the sodium, and that causes that threshold all up and down the axon to be reached, which means all the sodium gates are going to open, and they're going to go in one way. The reason why it doesn't go backwards is because of the inactivation gate on the sodium channel. Okay, well, let's not knock that over. They were about to knock over my camera. Yeah. All right. Hold on. So what happens, the reason why I can't go backwards is because that inactivation gate closes after you have your depolarization. And it remains closed for like a very short amount of time, but short enough that the sodium ions go in both directions on the axon. These gates back here won't open. They're closed by that inactivation gate. But these down here, that inactivation gate's not activated, right? It's not closing it. So the activation gate's all open, and then the axon, the it travels in one direction. Come on, ladies. Let's go. Come on. I will return with your questions. Come on. Let's go get it. Come on. Okay, come on. When I'm on the computer like that, not bother me. This is the ice we have. Here's what we have. Okay, what you need? I just got it for you. Here, look. Okay. Okay, do not bother me. Lily, this is the last one. I am sorry. I don't care what. Here, y'all work it out. Y'all work it out. Okay, all right. Yeah, there is a big fight because there's only one of the El Primo ice creams, and the other ones are not as El Primo. So I gave it to him, so let him work it out. All right, sorry about, or thanks for understanding. I understand what it's like now to be a single parent. I'm with the kids all the time now. Okay. All right. What do you mean by functional unit of a myofibril? That's a really good question. Sarcomere. So make sure you understand that. It's a sarcomere. Okay. Esmeralda, when you say you want us to explain the differences between a chemical and an electrical synapse, do you mean their defining characteristics or the process between each? That's a really good question, and I like how you were trying to ask specifically what we were looking at. And the reality is, when you think about the defining characteristics and the processes of each, they really go hand in hand. So the idea with an electrical synapse is, if I've got a synapse where two neurons are connecting, the electrical signal just goes from one synapse straight to the other, and the two neurons are actually connected to each other. And because they're connected, the chemical signal is actually sent through gap junctions in these. So there's no neurotransmitter, okay? And then the one with the chemical synapse, okay, there's a space between them, and that is the synaptic cleft. And what happens in this synapse, which could be between another neuron, it could be between like a gland, it could be between a muscle, that space, there's a chemical that's released from the end here, from the terminal, the axon terminal, to the other receptor cell, okay? And when the neuron releases the neurotransmitter, that's the chemical that's going over here, and it's going to bind to a ligand-gated receptor. And if it's like a muscle cell, the neurotransmitter is going to be acetylcholine, and the receptor is going to be a ligand-gated sodium channel, specifically one accepting an acetylcholine, which will open up the floodgates for this to create a local action potential inside the muscle cell. I hope that that's what I'm looking for on that one. Okay, Justin, can you Saeed, yeah. Can you explain the relationship between the plants and animals and multicellular or make multicellular? Very first part of this thing is what I went over. I went over it. Yeah, Willow, hi Lily. Thanks, ladies. Yeah, they're cute. They need attention. They're like little kids, Okay, Maddie, when asking about how the sarcomere contracts, are you looking for an explanation of only the sliding filament model or the whole three-phase contraction? Okay. Let me go to the specific question here. Let me guide you as best I can. But that's a really good way of you asking about sliding filament and the three-phase process. I'm going to tell you right now, I'm not really looking for the three-phase contraction process. What we're going to do is, let me see here. Let me make sure I get this right to you. Okay, so there are 3 stages, but think about it in terms of this way. You've got a muscle cell, a muscle cell, and it receives a signal from the motor neuron, and that signal is acetylcholine. That's the neurotransmitter. And the very first question is, how does the release of that neurotransmitter trigger the first part of the muscle contraction, the first part that's needed? And that is the release of the calcium ions from the sarcoplasmic reticulum. And if you say the terminal cisternae, totally fine, because the terminal cisternae is specifically the actual part of the sarcoplasmic reticulum that's releasing the calcium ions. So the part A, so it's all about muscle cells help animals move by contracting, which involves many steps and many unique structures. So we're going to, A is acetylcholine at the neuromuscular junction. How does that lead to the release of calcium ions in the sarcoplasm? So you need to talk about ligand-gated sodium channels at the motor end plate, creating a local action potential. Then you need to talk about voltage-gated sodium ion channels on the sarcolemma, creating the action potential that then goes down the T-tubules, the transverse tubules. You just call them T-tubules for this test. And then how that's going to open voltage-gated calcium channels on the terminal cisternae. That's part A. That's what you need to be able to explain that. Second, myosin cannot attach to actin in the sarcomere. So the sarcomere is the functional unit of a myofibril. And of course, it's made of actin and myosin. They slide past each other. This is the sliding filament theory. Okay, first, we're not talking about the power stroke here, but for this whole sliding filament theory to work, for the myosin heads to start to attach to the actin filaments in the first place, before you get the power stroke, for any of this to happen, you've got these regulatory proteins, troponin and tropomyosin. The tropomyosin is basically covering, it's covering up the active sites. So what you have to do is the calcium ions are going to bind to troponin to move the tropomyosin off those active sites. Okay, that's the importance of the calcium ions of when it can attach to the actin filament. So make sure you get those regulatory proteins right. It's all in my PowerPoints. That's the role of calcium. That's why that's the importance of the calcium ions and why that action potential needs to travel down those T-tubules so that the terminal cisternae will release the calcium ions. So basically you can regulate the sliding filament theory because it's the calcium that's going to affect the regulatory proteins that allow the myosin heads to actually attach to the actin filaments. And then the next one is describe this. Part C is describe the steps of the power stroke, starting with the attachment of ATP to the myosin head. So, okay, for it to even attach, for the myosin head to attach to the actin, we've already talked about the calcium ions. They're moving the regulatory proteins off those active sites. We start the power stroke with myosin already attached to the actin filaments, which means there's already calcium has been released, right? And then the power stroke is when ATP attaches, it comes off. It comes off. The ATP hydrolyzes to ADP plus ADP plus a phosphate. It cocks. It comes. The phosphates and the things come off. It reattaches. And then it does a whole power stroke and slides past it. So make sure that there's a great slide I have on that power stroke, starting with the myosin heads attached to the actin filaments. I think there's like 4 or 5 different steps. The ATP attaches, the ATP hydrolyzes, it comes off, and you repeat this over and over and over again. And you can continue to repeat it as long as there's calcium binding to those regulatory proteins, keeping the active sites open. Okay. So that is what I'm really looking for, Maddie. And I mean, that's the actual question right there. Those A, B, and C, from the start of acetylcholine at the neuromuscular junction, how does that cause a release of calcium ions into the sarcoplasm? What's the role of calcium ions in regulating when myosin attached to the actin? And describe the steps of the power stroke with the attachment starting with the attachment of ATP to the myosin. Okay, Maddie, great. I'm glad that makes sense. Yeah, you guys are asking all good questions tonight, and that was a really good one. All right. Yeah, and then it starts with the multiple choice here. There's an easy question. I was told that there was probably one or two too many essay questions, which I love asking essay questions. So I removed it. I added in an extra multiple choice. There is a question on here. It says, what is your favorite animal? Extinct, living, or mythical? The more creative, we might give you a little extra bonus in there on the TA discretion for if it's something really cool and not common. So, but no dogs, cats, or other domesticated animals or dolphins. Come on, there's more to life than I love my dog. Dolphins are really cool, but there's like 2 million species of animals. We can do better than dogs and cats. And then with the matching, okay, let me help you out with this matching really quick. Yes, Shy, definitely matching. Let me go over this really quick. The matching is going to be the structure with its function. And that is the different parts of like the CNS and the PNS, the peripheral nervous system, the central nervous system. So you need to know afferent neuron, somatic nervous system, sympathetic nervous system, parasympathetic nervous system, cerebellum, cerebrum, gray matter, white matter, diencephalon, and brainstem. And to be honest, I probably just pulled the definitions for these right out of the book and copied them as what they're. So there you go. Shy, I hope that helps out with. I just gave you all of them. Okay. One-way lungs allow for constant air exchange, whereas two-way lungs don't. A little confused on how they're different. Eric, yeah, you're right on the right path here. Think about when you breathe. 2 ways. Air goes in, and when that air goes in, it immediately diffuses into the blood, right? The oxygen diffuses into the blood. But because it's a 2-way lung, I've also got carbon dioxide being released from the blood into my lungs. So when I breathe in, there's already carbon dioxide in there. When I breathe out the carbon dioxide. But because I just breathed in air, oxygen, I also breathed it back out. So whatever didn't get absorbed all of a sudden also got exhaled. So it's not as efficient. It's pretty efficient. I mean, you can have big lungs and heart. Look at your dogs and just how constant a dog can run. I mean, they have big hearts, large lungs. They can run and run and run and run. Cats have small hearts and small lungs. That's why you don't ever need to walk a cat. And that's why they sprint and then they're done. The one-way lung is really cool. So when a bird or an archosaur, like a dinosaur, we don't have any dinosaur lungs that I know of, but we have bird lungs. And the bird, when you breathe in, it goes into an anterior air sac in the back. And then when it breathes out, it moves the air through the lung in one direction. So there's no back and forth. There's no exchange of carbon dioxide and oxygen, and you breathe in some oxygen and it gets exhaled right back out. No, that oxygen is going one way through the lungs, kind of like a fresh water going one way through the gills, right? But by going one way through the lung, it's way more efficient at extracting oxygen. And think about this: if you've ever seen like videos of birds flying really high, or gosh, you can go up to the Andes at 18,000 feet, and the field researchers are literally sucking wind, and there's hummingbirds, flying around no problem. There's other physiological differences, including differences in the hemoglobin and their affinity for oxygen. But those one-way lungs are very efficient at extracting more oxygen out of a thin atmosphere. Okay. Anybody else have questions? Man, you guys are asking good ones. You guys have been a fun class. Yeah. You're welcome. Yeah, anytime. I love it when you guys ask questions and try to, help me know what you're thinking. The 6 neuroglia. Yeah, I know them. It's not hard. Really make sure you know the difference between like Schwann cells and oligodendrocytes. That's a good one to know the difference of, because they're very important for forming the myelin sheaths, and the myelin sheaths help speed up the speed of an action potential. And in your peripheral nervous system, but you'll need to know the difference between central and peripheral nervous system. But in your peripheral nervous system, you've only got the Schwann cells. And in your central nervous system, that is done by the oligodendrocytes. So that's some things you'll definitely want to know for the multiple choice. Okay. Oh, there it is. Sharks can detect electromagnetic fields by... I'm not telling you the answer. You guys go look that up. What if sharks have to detect electromagnetic fields? And... oh, yeah, here's another thing you need to know about. Make sure... I talked a little bit about weight training. And of course, we all have our interests. We're all biased. I've been doing this for a very, very long time in my life, so it's very important to me. But exercise in general should be important for everybody. I mean, even just walking. I mean, like I haven't been in the gym in a while, and today I got my 11,000 steps in. I mean, I had to get up and go walk all the time and chase girls around and keep my step counter on me. They're watching Wildcats, by the way. So you need to know things like with training. Let's see here. Like what happens if you do endurance training? What kind of physiological changes might you see in your muscles with endurance training? So you need to know the difference between type 1, type 2. That's as broad as I'm going to get right here. But you do also need to know about myoglobin as well. And yeah, know about rigor mortis. Because in rigor mortis, because you don't produce ATP, because your membrane potentials will collapse inside your mitochondria, you're not producing copious amounts of ATP, so you can't remove calcium out of the sarcoplasm. The terminal cisternae have no ability to do active transport, so the calcium either leaks out of the terminal cisternae completely or can't be removed, so it's keeping the active sites of the actin open so the myosin heads stay attached, right? So because they stay attached and you're not producing any ATP to get them to pop off, your muscles become stiff and you have rigor mortis. Yeah, know the importance of the evolution of jaws. I love asking those evolution questions, even in a physiology class, because that evolution makes things make sense. Like, here's a little hint, jaws probably didn't evolve originally for bite force. It was probably gas exchange. That's why they gave up a gill. Okay, power stroke, action potentials, environmental stimuli, countercurrent exchange, different cellular respiration. Okay, that's good. Okay. Let's see. Okay. Justin, do we have to know how sharks drown? No, you don't. I don't think you need to know that for this test. I don't think I have a question on there. But the reason why they drown, and you'll learn this if you take a general vertebrate zoology class, is they do what's called ram ventilation. So they swim with their mouths open and they force water across their gills. So when they stop swimming, they stop moving water across their gills and they don't get the gas exchange they need, so they die. They drown. It's really crazy. Is electroreception limited to animals in water because water is a better conductor of electricity than air, or is there something that I'm missing? Maddie, you are spot on. That is why you and I and humans and land animals really don't have any form of ESP, because electrical fields don't travel very well through the air and we have no way of really detecting them, whereas they do travel through water very well. Okay, can you explain the chart for the afferent and efferent division one more time, please? Okay, I'm going to pull up the chart so that I can have it right in front of me. And let's see here. Okay. Yeah, I pulled this like the central nervous system. This, I think it's 4615 or something like that in your book. The textbooks like to update every few years and keep people buying new textbooks. Yeah, and then they don't really update all the advanced science, so that's pretty wild. Sorry. Okay, here it is. I'm on slide number 25, animal number 9. And if you want to pull this up, I don't really have a way. I couldn't get my Logitech to talk to YouTube tonight. Central nervous system, brain and spinal cord. You have a whole bunch of nerves that are detecting environmental stimuli and detecting stimuli inside your body. They're sensors. They're everywhere on you. They're in your stomach. They're in your gut. They're in your bladder. And then you have all the other stimuli of, like, you can feel when you're stretching your arms. You can feel that stretch. If you're working out, you can feel the burn. You can feel hot, cold temperature. These sensors relay information to your central nervous system. So all these sensors are part of your peripheral nervous system, everything that's outside of your brain and your spinal cord. That is the afferent nervous system. Afferent with an A. A-F. I'll say it. Oh, wait, hold on. I'll say it. Afferent division is transmitting is transmitting sensory information to your brain, both internally and externally. Got to pee? That's your afferent nerve division. You've actually got a bladder, and when it expands, it actually creates a stress on those cells, and it trips a signal. You know, it opens up some voltage gated, or not voltage gated. It opens up. Oh gosh, I forgot the name of it. There's actually a channel that will open up based on pressure gated, I guess, and it's a signal. You got to go pee. So that is part of the afferent nervous system. It goes up to your brain, your central nervous system. Of course, it goes through your brain stem, then it goes through the diencephalon, which will then determine where these signals go. That's your, okay, if it's your bladder, it goes here. If it's from your eyes, it goes there. If it's from sense or smell, it goes to your, smell goes to your olfactory bulbs. Okay, now your stomach is hungry and you smell pizza, right? Okay, this is your afferent nervous system going, you're hungry, you need to eat, and your blood sugar levels are getting low, and you smell pizza, and you see it in the kitchen out there. So what's going to happen now is your central nervous system is going to take in all these stimuli. It's going to process it. You're going to say, you can go eat that pizza. You can go eat that slice. So now it's going to send a signal down what's called your efferent nervous system. All right? Efferent division, sorry. It's going to send this down your efferent division. And there are two parts of the efferent division, both the somatic and the autonomic. Okay, let me write these down for you. Autonomic. Okay, so the somatic nervous system, this is me doing all my hand gestures, right? I'm going to get up. I'm going to go walk into the kitchen to go get the pizza. That is the somatic nervous system. That's the efferent nervous system. So my brain is sending signals down my motor neurons, right? That basically is going to say, get up and go walk. That is voluntary. On the way there, my 2-year-old and 5-year-old ambush me and jump out and scare me. I get all, I jump, adrenaline release. Okay, now I have an autonomic nervous system. That adrenaline release, because they decided to jump out and ambush me on the way to go get my pizza. Okay, I release adrenaline, right? And then what happens with these two divisions, okay, if you remember, I used the get to the chopper. So the sympathetic nervous system, this is the adrenaline rush. This is, okay, my pupils are going to dilate. I'm going to shut down, digestion. I'm going to release glucose into my body for the flight. I'm going to dilate my blood vessels. My heart rate's going to shoot up. My breathing rate's going to shoot up as I get to the chopper. Fight, flight. Actually, I'm not going to fight my girls, right? Probably jump. But that's your flight or flight response. Then the other one is the parasympathetic nervous system. Once I realize Lily is pranking me, actually she doesn't prank me. It's my 2-year-old Willow. She starts pranking me. What happens next is you return back to a more resting state. My blood vessels are going to dilate. I'm going to start up digestion again. My mouth is going to start to salivate because I'm getting ready to eat. So that is in a big way that chart of the nervous system, right? Afferent is all your sensory nerves going to your CNS, your central nervous system. It processes it. It sends what you're going to do back down through the efferent division, which can be voluntary, which is somatic, that's you moving around, or it can be involuntary, which is both the parasympathetic and the sympathetic nervous system. Okay. All right. Alicia, I hope that works. Elliot, why is the sarcomere dark? So because both dark and thin filaments are on it. You nailed it. That's exactly correct. That's why it's dark. And then the light one is light simply because that's basically all you have is a little bit of elastin and actin filaments. So good job. Maria Nava, when asking about the two basic muscle types in humans, are you referring to the two muscle fibers? Yeah, I am actually. And in fact, I'm sorry if I caused any confusion because there's cardiac muscle, smooth muscle, and skeletal muscle. All the test questions will be about skeletal muscle. I'm not asking anything about smooth or cardiac. I know there's a lot of guys taking AMP or students taking AMP. They're like, oh yeah, we need to learn that for that, but we don't really have a lot of time to get into those details. So I focused on skeletal muscle. So if I'm going the two basic types yeah, type 1, type 2. It's much more complicated than that, even in humans. And also know that in the animal kingdom, it's more complicated as well. But we just, let's just focus on something we can all use as a take-home message at the end of the semester. All right. 40 minutes in, we did good, guys, ladies. We did good. Any other questions? Thanks for the thumbs up, by the way. I love it, man. It makes me feel good for doing this. Yeah. Good luck on the test tomorrow. I don't think you're going to need it. I kind of helped you out here. I want to see everybody do well. Remember, on these questions, if it's 5 points, details matter. Thanks for the thumbs up. Details matter, right? They really do. Like, can you tell the story? Can you tell it well? Superficial knowledge on some of these larger essay questions, yeah, we're trying to get you to think, about the more details of a complex system. And like I told section 2, you might say, well, I'll never learn sliding filament theory ever again. I don't really need this, or it's boring, or something like that. Or there's so many parts to a muscle fiber, and in all these different moving parts. And you really need to know that, because I worded a lot of the multiple choice to make sure that you know those terminology to check it. This is also a life skill, right? I mean, learning how to memorize a bunch of different terms that you're very unfamiliar with. I mean, sarcolemma, axonlemma, sarcoplasmic reticulum, terminal cisternae, triads, transverse tubules, myofibrils, sarcomeres, Z-lines. These can make your head swim. But if it's like the Karate Kid, wax on, wax off. He learned a skill that he could use elsewhere in life. And if you can learn this, like how a muscle contracts, and really learn it well, then you've trained yourself how to learn something complicated. Will you know this again sometime in your life? Maybe, maybe not. I find it fascinating. I enjoy learning this stuff just because I find it utterly fascinating. Some of you may not, and that's okay. But this is like you training your brain to learn a lot of terms and a lot of moving parts to tell a complex story. And if you can do this here, guess what? You can do it elsewhere. You can do it in your other classes. You can do it on a job. That's why they like college graduates, because college graduates, you're taught to do something like this. Okay, that's me on my little, giving a little prep talk there. You guys are going to do it. You've done really well this semester. As expected, test 3 had a much higher grade. Oh, I'm at 23. Yeah, you guys did good. Naya said she kept a note of every time I mentioned pizza. Yeah, we're at 23. Also, thank you for everything. You are so welcome. You guys have been a really wonderful class, both sections. I've really enjoyed interacting with you this semester. And hopefully, I will see some of you again this fall in astrobiology. That class is really fun. We're going to talk about aliens in the semester. And hey, if you're in astrobiology, or get somebody to how many times does Kennedy say, does Tom say pizza? I was actually too old for Teenage Mutant Ninja Turtles when they came out in the 90s. I was like in my late teens, and it was a big rage. Yeah, the raves were when I was in college. All right. Any other questions? Are we all good? All right. I'm signing off. I will see you guys tomorrow. Like I said, you got this. You're going to do well. I'm really rooting for you. I know you're going to do well. All right. You guys are welcome. Yeah. Anytime. Oh, thank you so much for all the kind words. It's really nice to see all of this. Like all other influencers here, we as a professor, even we live to see the positive feedback. So thank you so much for everything. And I'll see you tomorrow. I'm going to sign off.